Hello everyone. So today in this video we are going to solve the CPET 2020 Zoology paper. So I'm dividing this entire video into two parts. This is the part 1 of this video, and in this part we will be solving from question number 1 to 35. And in the second part of the video we will solve from question number 36 to 70. So without any further delay, let's get started. First of all, question number 1, that is, amoebic dysentery is caused by, the options are Amoeba proteus, Entamoeba histolytica, third is Plasmodium vivax, fourth is Taenia solium. The answer for this will be Entamoeba histolytica. Okay, Plasmodium vivax, we all know it causes malaria. Taenia solium is called teniasis. So, what happens in amoebic dysentery particularly? This Entamoeba histolytica is transmitted through contaminated food or contaminated water, and if this dysentery is particularly not treated in time, it may also prove to be fatal. Okay. Third question. In sponges, the cell surrounding the osculum is, answer for this is B, myocyte. How is myocyte responsible for surrounding the osculum? Let's see a closer structure of sponge for this purpose. This is the osculum, that is opening of the sponge, and this opening is responsible for the entry of water, incurrent water, and the removal of water, that is excurrent water. Okay, excurrent and incurrent channel, it is responsible for this. And since it is regulating the inflow and outflow of water, this particular opening should have some structures that should be able to contract. Okay, that should be able to contract and relax. For this purpose, we need something like muscles. Muscles have the ability to contract and relax. So these are myocytes. These act like just muscle cells in case of sponges. Fourth question. Conchology is the study of coelentrates, no. Study of coelentrates is nidology. Okay. Study of mollusca is again what? It is malacology. And study of shells of mollusk, yes, this is conchology. So answer will be question number 4's answer will be C. That is shell of mollusk is called conchology. This is the whole diagram of I've given it here for the purpose of ease of understanding. What are the different cells present in case of sponges and what are their functions? Okay, try to remember particularly the choanocyte cells. It is frequently asked about the choanocyte cells and amoebocyte cells. These are the frequently asked questions from sponges. Okay, next. Syncytial epidermis is found in case of Ascaris. Okay. Question number 6. Aristotle's lalton is present in case of sea urchin. Okay, sea urchin. What is Aristotle laltons? It is basically a teeth apparatus. Okay, it is a teeth apparatus. It is present in case of sea urchin. Next, in earthworm, ovary is situated in which segment? Answer will be 13th segment. Here in the picture we can see ovary is present where? In the 13th segment. Remember students, earthworm is what? Earthworm is a hermaphrodite. Earthworm is hermaphrodite. Okay, hermaphrodite. Hermaphrodite means a single individual has both the gender. That is, it can have the male gender as well as the female gender. A single individual. As you can see, this is a single earthworm and it is having testis over here in the 10th segment. Also, there is ovary in the 13th segment. Okay. Next question. Question number 8. In cephalochordates, gill slits opens into, answer is atrium. These are the pharyngeal gill slits and they are opening where? To this atrium. Next question. Mammals like reptiles belongs to which subclass? Anapsid? No. Urapsid? No. Parapsid? No. Answer will be synapsid. How synapsid? Let's look at the brief outline about class Reptilia. So these are the 5 subclasses present in case of Reptilia. Remember, out of these 5 subclasses, only 3 classes are extant. 3 classes are extant and 2 classes are extant or living. Okay, 2 classes, that is Anapsida and Diapsida. These 2 are living classes. Other 3 are extant. And out of these extant classes, Synapsida is the subclass. Synapsida is the subclass which are mammals like reptiles. Next question. Question number 10. So fossil records of Archaeopteryx have been discovered from, the answer will be Germany. It was discovered from Germany. Archaeopteryx is again a connecting link between what? Aves and reptilia. Okay, aves and reptilia. Largest living land animals are included in the suborder. Okay, if you don't know the names of the suborder, don't worry and don't get panic. Simply do what? Decode the question first. Question is asking what? Largest living land animal. We know largest living land animal is what? It is elephant. It is elephant. Now look at the options. Now they can seem easier. Now look, option number 3, that is Proboscidea. Okay, option number 3, that is Proboscidea, will be the right answer. Question number 12. Okay, this is the easiest question of the entire paper. The driving force of an ecosystem is, answer is solar energy. Solar energy is the driving force of entire ecosystem. The term ecotype was coined by, answer is option number A. What is ecotype? You need to remember. Ecotype, I'm explaining it. Let's understand it with a short example of polar bear. You must have heard about polar bear and normal black bear. Bear is found in polar regions of Earth. These are white in color, right? It's white in color. You must have seen in the Discovery Channel at least. And next one is the black bear. Though these polar bear and black bear, they belong to the same species. Okay, they belong to same species. But somehow they are genetically different. Somehow they are genetically different and occupy different geographical areas, right? So that is why these are known as ecotypes. So what are ecotypes? Ecotypes are members of the same species which are genetically different. So next term is biocoinosis is was proposed by, answer will be Carl Mobius. It is Kark over here. It will not be Carl. It is Carl. Okay, Carl Mobius. Answer option C is the correct answer. Next, question number 15. Which of the following is not a limiting factor for photosynthesis? Not a limiting factor for photosynthesis. Okay, what is again a limiting factor? First of all, let's understand. Limiting factor is something that is present in minimum. Okay, when a reaction is occurring, in any reaction, the factor which is present in minimum, okay, minimum, it is called limiting factor. Okay, now let's have a look on the equation of photosynthesis. This is the equation of photosynthesis. We have carbon dioxide, water that gives rise to glucose and oxygen. And this reaction occurs in the presence of light, that is sunlight, and chlorophyll. So what can be a limiting factor over here? Okay, what are the reactants we need? We need carbon dioxide, yes. We need chlorophyll, yes. We need light. Do we need oxygen for the reaction to occur? No. Oxygen is a product over here. Here we can see, like, oxygen is the product. So the limiting, it is not a limiting. Which of the following is not a limiting factor? The answer will be our option number 1, that is oxygen. Okay, this is the answer. Next. Receptor-mediated endocytosis from plasma membrane requires which of the following coat protein? Remember, the coat protein which is required for endocytosis. Endocytosis is what? Engulfing. Okay, engulfing action of the cell is called endocytosis. Engulfing action of the cell is called endocytosis. For endocytosis purpose, what is needed? A coat protein is needed, and this coat protein is called clathrin protein. Here are different types of endocytosis. I have given a diagram over here. Here you can see this is clathrin mediated endocytosis. Number 2, this one is clathrin mediated endocytosis. Here the proteins, clathrin proteins are present. Okay, clathrin proteins are present around the vesicles. Okay. Next question. The protein that is responsible for sliding of outer microtubule doublets against one another to produce ciliary binding is, answer for this will be dynein. Okay, remember there are two types of motor proteins which helps in the movement of intracellular organelles. Okay, what are these? Dynein and kinesin. Another protein is kinesin. Okay, the protein is plus n directed, then what protein comes into action? Kinesin. When it is minus n directed, then dynein comes into action. So for microtubule transport, any food vesicle or anything that is getting transported in the cell, it is transported with the help of these two proteins, kinesin and dynein. Remember, if it is minus end, then it is dynein. If it is plus end, then it is kinesin. Okay, next question number 18. Which of the following protein targeting mechanism is common for mitochondria and chloroplast organelles? Okay, which of the following protein targeting mechanism is common for mitochondria and chloroplast organelles? Protein targeting mechanism. We all know central dogma of cells. What central dogma says? Information is transmitted from where? From DNA to RNA. Okay, then from RNA to it is transmitted as proteins. Okay, it is converted into proteins. So this protein by the process is what we call it, translation. We call this process as translation. And DNA to RNA, that process is called transcription. Okay, transcription. So how the protein is transported into the organelles called mitochondria and chloroplast. Here we can see in the diagram. In the nucleus, there is present DNA. From DNA, mRNA is synthesized. From mRNA, again, proteins are synthesized here. Protein is formed. And this protein, after the process of translation, protein is formed after the process of translation. Right. After the process of translation, this protein is incorporated into the organelles. Okay. So how it is getting incorporated? Post-translationally. Post means after. After the process of translation, protein is getting incorporated. So answer will be B. Then question number 19. Cell cycle is controlled by what? Students, remember, cell cycle is controlled by different proteins, and these proteins are called cyclins. Okay, these proteins are called cyclins. Cyclins are what? These are substances which get synthesized in particular phase of cell cycle. We know the cell cycle has different phases, right? Like there is G1 phase, then we have S phase, then we have G2 phase, right? Then we have M phase. Okay, these are the different phases of cell cycle. So during each phase, different cyclins are synthesized. Okay, different cyclins are synthesized and they get activated at particular phase. Like in the S phase, what is activated? Cyclin A is activated. Okay, in the G2 phase, that is cyclin B is getting activated. Okay, B and A both have role in G2 phase. So the cell cycle is controlled because of these cyclins. Okay, so let's have a look at the options. Proteolysis of cyclin. Proteolysis means breakdown. Okay, breakdown of cyclin dependent kinase. Cyclin dependent kinase is another enzyme. Okay, breakdown of CDKs. No, this option is incorrect. Phosphorylation of cyclin. Yes, cyclins, of course. Option number B will be correct. Phosphorylation of cyclin. Phosphorylation does what? Activates these cyclins. Okay, phosphorylation activates cyclin, and this is how it controls the cell cycle. Next question, question number 20. Which of the following biochemical reaction is most commonly utilized by living cells to propagate intracellular signals? Just now we saw what it will be. Option number B, that is phosphorylation. For any transmission of intracellular signal, mostly we use the process of phosphorylation. Next, question number 21. Isotopes used for proving semi-conservative DNA replication of DNA are option number 1, N14 and 31. No, this is not correct. Option number B, N14 and N15. Yes, this is the correct. Okay. You must have remembered the test in which we use cesium fluoride gradient. Cesium fluoride gradient to prove the semi-conservative nature of DNA. Like we got two bands over here. First the N14 is here, N15 is here. Okay. When we use both the bands, we got an intermediate lining, which proved that DNA is semi-conservative in nature. Next, question number 22. In prokaryotes, the lagging primers are removed by. Options are A, 3' to 5' exonuclease activity. No. DNA ligase. No, absolutely not. DNA ligase is for joining. Right? Joining. It is no. DNA polymerase 1. Yes. Option number C will be the answer. Lagging primers are removed by DNA polymerase 1. Okay. Next, question number 23. When human genome draft sequence was released, which of the following observations was least expected? Okay. When human genome draft, the human genome sequencing was done, when human genome sequencing project was done over, what we expected was the humans have lots of genes. We have numerous genes. There must be billions and billions of DNA sequence present in our DNA, right? But this was not there. Okay, we have merely similar DNA sequences as there were in C. elegans. As there were in C. elegans. Okay, nearly it was same. But what we had that was different was amount of repetitive DNA. So amount of repetitive DNA was more in case of human beings than expected. So this was the surprising thing which was concluded after the sequencing was over of human genome sequence. Sorry. Okay. Then question number 24. What is photoreactivation? Photoreactivation does what? Repairs dimers in DNA using endonuclease. Yes, absolutely. Option number A is right. Position effects results in. What is position effects? Okay, effects. Let's understand this. Suppose this is a chromosome. Okay, chromosome had different segments over here. It has different segments. If some segment is getting mutated, okay, mutation also can be of different types. If it is getting inversion, means a position is changing. What will happen? Ultimately, the genome will be changed. Suppose this is the one number chromosome. It is getting inverted and it is converting into three number. So 1 and 3, are they similar right now? No, they are different. And this difference is caused by inversion. So what is happening over here? The position of a sequence is getting changed. Okay, the position is getting changed. So answer will be C, that is inversion. Okay, question number 26. The perineurium is the connective tissue layer. Perineurium is the connective tissue layer surrounding the fascicles of axon. Option number D will be the correct answer. How it is correct? Let's understand. Suppose this is a nerve fiber. Okay, this is the nerve fiber. The nerve fiber, the entire nerve fiber is again made up of different fascicles, small fascicles like this. And each fascicle has again the myelinated or unmyelinated nerve fibers. It is containing the myelinated and unmyelinated nerve fiber. So what we have the spinal nerve over here. Spinal nerve is having small fascicles. Okay. And in these fascicles, okay, in these fascicles, what happens? In these fascicles are present what? Our nerve fibers that can be either myelinated or unmyelinated. So whole nerve is surrounding by a layer that is called epineurium. This layer is called epineurium. Epineurium. And these fascicles are surrounded by a layer that is called perineurium. Neurium. And again, these nerve fibers are surrounded by a layer which is called endoneurium. Okay, clear about the connective tissues layer which are surrounding the nerve. The whole nerve bundle is surrounded by epineurium. The fascicle is surrounded by perineurium, and the individual neurons are surrounded by what? Endoneurium. So answer for this, perineurium is the connective tissue layer that is surrounding the fascicles of the axons in the PNS, peripheral nervous system. Next, question number 27. The most important factor in determining percent oxygen saturation of hemoglobin is option number A, that is partial pressure of oxygen. Okay, partial pressure of oxygen. Remember, as we increase the partial pressure of oxygen, okay, we will find an increase in percentage saturation of hemoglobin of oxygen in hemoglobin. Percentage saturation of oxygen in hemoglobin. Okay, here is a graph I have kept. Look, as we are increasing the partial pressure, Similarly, the percentage saturation is also increasing. Okay. So option number A will be the right answer. That is partial pressure of oxygen. Question number 28. Reabsorption of chloride ions from glomerular filtrate in the kidney tubules is carried out by. Always remember, when it is about the transport of ions, it is done by active transport. Okay, active transport, that is with the expenditure of energy in the form of ATP. Okay, so answer will be C. Question number 29. Gastric glands whose absence would lead to pernicious anemia is. Okay, look, anemia can be of different types. Pernicious anemia, why it is caused? Pernicious anemia is caused due to deficiency of vitamin B12. Okay, vitamin B12. Particularly, because of the absence of vitamin B12, pernicious anemia is caused. Look, which gastric gland cell is responsible for this? Remember, here answer will be your option number D, that is parietal cell. Parietal cell is otherwise also known as oxyntic cell. Oxyntic cells. And what is the function of oxyntic cell? They secrete HCl, hydrochloric acid, and intrinsic factor. Intrinsic factor. What is the role of intrinsic factor? Intrinsic factor facilitates the absorption of vitamin B12. Intrinsic factors, what does? It helps in absorption. Absorption of what? Absorption of vitamin B12. Right. So if this parietal cell is having some defect, okay, if we don't have parietal cells, proper parietal cells, what will happen? We won't be able to absorb what? Vitamin B12. And what will happen? Then the condition will be caused, that is pernicious anemia. Coming to question number 30. All the hormones of adrenal cortex are synthesized from. Remember, adrenal cortex, all the hormones which are synthesized from adrenal cortex are steroid in nature. Steroid in nature. So, steroid hormones are always synthesized from what? From cholesterol. Okay, answer C. Next question, question number 31. The primary target organ of aldosterone action is, answer will be kidney. So kidney is the primary target organ for the action of aldosterone. So what aldosterone does? Aldosterone helps in regulation of salt, okay, salt, water balance, and blood pressure. Okay, volume also, blood volume and blood pressure. Blood pressure. It helps in controlling what? Salt balance, blood volume and blood pressure. Okay, and for these action, what is needed? Aldosterone is needed, and the action site for aldosterone is kidneys. What it does, it increases the sodium absorption. Okay, it increases the sodium absorption and potassium secretion. Sodium reabsorption and potassium secretion. Next, question number 32. The bacteriophage M13 contains as its genetic material what? It will be single-stranded DNA. Option number C will be the correct answer, that is single-stranded DNA. So next question is question number 33. That is, what is the number of hydrogen bonds? Okay, what is the number of hydrogen bonds in bDNA of 1000 base pairs? Okay, with the proportion of CAT in one of the strand corresponding to 60%, 30%, and 10%. Okay, what is the question asking? It is asking the number of hydrogen bonds. What is the number of hydrogen bonds we need to find out? Okay, we are having how much base pairs? We are having 1000 base pairs. If there is 1000 base pairs, means what? One strand is having 1000 bases and the second strand is also having 1000 bases. Right? What is given over here? It is given that in one of the strands, okay, in one of the strands, the base pairs are present, bases are present, C bases are present, cytosine bases are 60%, okay, in one of the strands, and adenine bases are 30%, and thymine bases are again 10%. Okay, if 60% of 1000 will be how much? 60 by 100 into 1000, that is 600. Similarly, 30% will be how much? 30% will be 300 and 10% will be how much? 100. So we need to find out the number of hydrogen bonds we are having. Okay, what is the number of hydrogen bond? Cytosine will form how much hydrogen bond? Cytosine will form 3 hydrogen bond. So 600 into 3, that is equal to 1800. Similarly, adenine will form how much hydrogen bonds? It will form 2 hydrogen bonds. So we have 300 into 2, that is how much? 600, right? Next we have how much? We have thymine. Okay, thymine will also form 2 hydrogen bonds. Okay, so we have 100 into 2, that is equal to 200, like this. So what will the total number of hydrogen bonds? We have it 2000 plus 600, that is 2600. Okay, this will be the answer. Option number A, 2600 is the right answer. Next, question number 34. In Sanger's method of DNA sequencing, the growing DNA chains are terminated because the growing chains are terminated. What happens in Sanger's method of sequencing? It is based on the presence of 3' OH group. Okay, we know DNA synthesis always occurs from which direction? 5' to 3' direction. So it is always dependent that we have a 3' OH group, right? We should have a 3' OH group, and that is how chain synthesis occurs, right? So how the chain is terminated? Chain is terminated because DNA polymerase is not very processive. No, absolutely no. There is no problem in DNA polymerase. DNA polymerase is absolutely processive. Radioactive nucleotide is incorporated. No. This is the method in which we need to use radioactive nucleotide. So this is not the answer. Okay, substrate becoming limited. No, substrate is absolutely present. A phosphodiester bond cannot be formed. Yes, this is the reason. A phosphodiester bond cannot be formed from the 3' OH group. Okay, 3' OH group was forming what? Phosphodiester bond to form the whole length of DNA that was synthesized from 5' to 3' direction. So when this phosphodiester bond is getting stopped, this bond is not formed, what will happen? There will be termination of DNA chain synthesis. So option number D will be the right answer. That is phosphodiester bond cannot be made. Next question. Glucose and galactose are two isomeric monosaccharide known as what? Answer for this will be epimers. Okay, what are epimers again? Let's have a look. Epimers are those saccharides, okay, those saccharides, polysaccharides particularly, which differ in one carbon atom. Okay, isomers are what? Isomers have same molecular formula, different structural formula. Okay, we can see the formula of glucose. This is the formula of D-glucose. This is the formula of D-galactose. Both have the same molecular formula. Both have the same number of carbon, hydrogen, and oxygen. Okay, but where they differ? They differ in a single carbon position. That is in the C4 position. You can compare both these structures, glucose and galactose. You can see everything is similar. Here is one hydrogen, here is also one hydrogen, here is OH, here is OH. Similarly, everything is same except this fourth position. They are epimers because of this fourth position, which are differing. Okay. So this was all about the 35 questions in this particular video. In the next video, that is part 2 of this particular paper, we will be covering from question number 36 to question number 70. I hope you like the video. Thank you all.